Good day, this is Professor Will, CBMD PhD, and today is Monday, November 29, 2021. It is 8:50 and 52 seconds, p.m. Eastern Standard Time, and today's research, humans and chimpanzees. So let's hop to it here. What makes us human? The answer may be found in overlooked DNA. Date October 8, 2021. Source, Lund University. Our DNA is very similar to that of the chimpanzee, which in evolutionary terms is our closest living relative. Stem cell researchers have now found a previously overlooked part of our DNA, so-called non-coded DNA, that appears to contribute to a difference, which despite all our similarities may explain why our brains work differently. The chimpanzee is our closest living relative in evolutionary terms, as research suggests our kinship derives from a common ancestor about 5 to 6 million years ago. Our evolutionary paths separated, leading to the chimpanzee of today and Homo sapiens, humankind in the 21st century. In a study, stem cell researchers examine what it is in our DNA that makes human and chimpanzee brains different, and they have found answers. Instead of studying living humans and chimpanzees, researchers used stem cells grown in a lab. The stem cells were reprogrammed from skin cells by their partners in Germany, the USA, and Japan. Then they examined the stem cells that they had developed into brain cells. And using the stem cells, the researchers specifically grew brain cells from humans and chimpanzees and compared the two cell types. The researchers then found that humans and chimpanzees use a part of their DNA in different ways, which appears to play a considerable role in the development of our brains. The part of our DNA identified as different was unexpected. It was so-called structural variants in DNA that were previously called junk DNA, a long repetitive DNA string which has long been deemed to have no function. And previously researchers have looked for answers in parts of the DNA where the protein-producing genes are which only make up about 2% of our entire DNA, and examined the proteins themselves to find examples of differences. The new findings thus indicate that the differences appear to lie outside the protein-coding genes in what has been labeled as junk DNA, which was thought to have no function and which constitutes the majority of our DNA. And this suggests that the basis for the human brain's evolution are genetic mechanisms that are probably a lot more complex than previously thought, as it was supposed that the answer was in those 2% of the genetic DNA. The research results indicate that what has been significant for the brain's development is instead perhaps hidden in the overlooked 98%, which appears to be important, and this is a surprising finding. Researchers discovered that specialized cells can be reprogrammed and developed into all types of body tissues. Researchers gazed into brain cells. Without this technique, it would not have been possible to study the differences between humans and chimpanzees using ethically defensible methods. And why did the researchers want to investigate the difference between humans and chimpanzees? Researchers believe that the brain is the key to understanding what it is that makes humans human. And how did it come about that humans can use their brain in such a way that they can build societies, educate their children, and develop advanced technology? It is fascinating. But further research on the 2% of coded DNA, research may now be forced to delve deeper into all 100%, a considerably more complicated task for research. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.